All right. I'm so sorry, guys. This is coming out so late. I had a really chaotic week, but I'm hoping this will still be helpful. And if you have any questions, as always, feel free to reach out. I'm more than happy to do office hours with you guys on Monday if anybody needs to, or if anybody wants to just email me questions. I'm happy to help. But besides that, let's jump right into your last review. All right, so you guys talked about metabolism, respiration, all that fun stuff. So I know this is so much information and there's so many reactions, and I remember how overwhelming metabolism can be. But I'm going to try to break this down as much as I can, and hopefully this will help. So, what is glycolysis? So essentially, it is the splitting of glucose and the overall breakdown of glucose into 2 ATP, 2 NADH, and 2 pyruvate. Glucose is a very versatile energy storage molecule. This is why glucose is used for this. It can be easily stored, and that energy storage can also easily be reversed and can be used for energy. So if you guys remember glycogen from when we talked about polymers and all that, so that was the storage form of glucose, and glucose is very rich in energy. And besides that, it is also a pretty universal pathway. It can be found in pretty much any mammal and a lot of other organisms. The full breakdown of glucose can yield up to 2,840 kilojoules per mole of free energy. So yield means that we get that free energy, so it is a negative value, just as a quick reminder. And glycolysis itself does not require oxygen. However, if we want to get that full free energy, and if we want to produce a lot of ATP, then we need to enter oxidative phosphorylation, for which we do require oxygen. But for glycolysis itself, we do not require oxygen. All right, quick overview of glycolysis. It is broken down into 2 steps. I'm realizing that this is not the most ideal picture. I apologize for that. But in the first phase, we invest energy. So that's why it's called the investment phase. It requires 2 ATP because we need to prime our glucose molecule. These are steps 1 through 5. And then the second phase, this is called a payoff, we produce energy and our reduced cofactors, which we can then later on use to yield even more energy. But in that second phase, we obtain 4 ATP and 2 NADH per molecule of glucose. So overall, that comes out to 2 ATP and 2 NADH. Because we consume 2 ATP, we make 4. Overall, we get 2. And glycolysis is a process of 10 enzyme catalyzed steps, and it occurs in the cytosol. So the first step, like I already said, is an activation step. We activate glucose via hexokinase. We add a phosphate group to the glucose molecule. The phosphate comes from a molecule of ATP. So we need to break down that ATP to add that phosphate group onto the C6 molecule of glucose. We also have a cofactor, magnesium, which helps stabilize that negative charge in the ATP molecule. So that is an important cofactor that hexokinase requires for this reaction. So now we have our activated glucose molecule, glucose 6-phosphate. This step is metabolically irreversible because we produce a lot of energy, and we don't produce energy, we consume energy, and we invest ATP. Our next step is the conversion of glucose to fructose 6-phosphate, which is catalyzed by phosphoglucose isomerase. So essentially, it turns that glucose 6-phosphate into a fructose 6-phosphate. So it just changes the arrangement of the molecule. Pretty simple. our third step is another activation step. It's the phosphorylation of fructose 6-phosphate to fructose 1,6 biphosphate. So we add a second phosphate group, and this is also our first committed step, which is catalyzed by phosphofructokinase 1. Again, it's our first committed step. This is where we commit to glycolysis, because once we have this fructose 1,6 bisphosphate molecule, we need to continue with glycolysis. This step is highly regulated because of this. So this is where a lot of feedback inhibition comes into play. All right, our next step is the cleavage of fructose 1,6 biphosphate by aldolase. So we have our C6 body, we have a molecule with 6 carbons, and that is split into 2 molecules with each 3 carbons. So from one C6 body, we go into 2 C3 bodies. One, we have dihydroxyacetone phosphate, and then we have glyceraldehyde 3-phosphate, so G3P, which is a little bit easier to say. Dihydroxyacetone phosphate cannot continue in the pathway, so it needs to be isomerized to glyceraldehyde 3-phosphate. So we can interconvert between dihydroxyacetone phosphate and G3P. The cleavage from between the C3 and C4, so from the C6 body into 2 C3 bodies, requires a carbonyl group. So we need to open up that fructose ring to get a linear molecule, and then it binds to the enzyme active site, forms a tetrahedral intermediate, and then it rearranges to get that carbonyl group, and then we split to get those 2 molecules. All right, our fifth step and the last step of the first phase is the interconversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, because, like I already said, we cannot utilize dihydroxyacetone phosphate in glycolysis, so we need to convert it. This isomerase that catalyzes this reaction, the triose phosphate isomerase, is pretty much a perfect enzyme because it has reached catalytic perfection. If you guys remember from when we talked about enzymes and how they can operate at pretty much a perfect speed. All right, and then we move on to the second phase of glycolysis. Remember that we now have 2 C3 molecules, so everything that we talk about from now on happens twice for each molecule of glucose. So we move on with our glyceraldehyde 3-phosphate, which then gets oxidized to 1,3-biphosphoglycerate. So we add another phosphate group on here, so we activate our molecule. So it looks like we're adding energy, and overall we do. So our free energy is positive, free energy change. And this step is possible because we are coupling it to the next step that follows, which makes this possible. And in the progress of this oxidation reaction, we reduce a molecule of NADH plus to NADH and a proton, which is super important for the oxidative phosphorylation later on. Okay, this is our step 7, and this is a step that is coupled to the previous step. We produce a lot of energy, or we get a lot of free energy in this step, which makes the step previous possible. So we have 1,3-biphosphoglycerate, which transfers a phosphate group, which then releases energy. So phosphoglycerate kinase catalyzes this step, and our product is 3-phosphoglycerate. And overall, the free energy change here is negative, which means it's a spontaneous reaction. Step 8 is the conversion of 3-phosphoglycerate to 2-phosphoglycerate via the phosphoglycerate mutase. So essentially, we are moving our phosphate group from the C3 carbon to the C2 carbon. In this step, we bind our product, our 3-phosphoglycerate, binds to the active site of the phosphoglycerate mutase, which has a phosphate group attached to it. So it is an enzymatic activation. And our intermediate is the 2,3-BPG, or 2,3-biphosphoglycerate, which that should ring a bell. If you guys remember from the oxygen binding curve and hemoglobin, that was a molecule that was pretty important. But our final molecule here, our product from this reaction, is 2-phosphoglycerate. So a reaction catalyzed by mutase. And then step 9 is the dehydration of 2-phosphoglycerate to phosphoenolpyruvate via enolase, and we release a molecule of water. And then our last step, in which we produce one more molecule of ATP, this is where we generate energy again, is our reaction from phosphoenolpyruvate to pyruvate via pyruvate kinase. So we now have our reduced molecule, pyruvate. Well, we have 2 molecules of pyruvate per 1 molecule of glucose, and we have produced a lot of free energy. Well, not a lot, but we have produced some free energy. Overall, 2 molecules of ATP, 2 molecules of NADH. And now also think about all the steps that can be regulated in this glycolysis reaction. In which steps do we release a lot of free energy? Which steps are the committed steps? Which steps are irreversible? And why should they need to be regulated? So that's an important point to consider. So now, how can we move on with pyruvate? We can actually have quite a lot of different fates. We can either move on to oxidative phosphorylation and the citric acid cycle, which we will talk about in just a second. This is only possible if we have oxygen available. We can move pyruvate into other anabolic pathways. For example, we can turn it into oxaloacetate, which can then be turned into different amino acids. Or if we do not have oxygen present, but we still need to somehow continue to generate energy, then we move on with fermentation. So in our muscle cells, that would be lactic acid fermentation, or in yeast, it is alcoholic fermentation. And the reason why we need to do this is because we need to regenerate our NAD+ molecules so that glycolysis can continue as usual, and we can still generate energy. So in this step, well, in the lactic fermentation or in alcoholic fermentation, we actually do not produce any energy, but we did have those 2 ATP that we produced in the first 2 steps of glycolysis. And in order for that to still continue, we need to regenerate our molecules of NAD+. So in lactic acid fermentation, we have pyruvate, which transfers its hydrogen, well, which takes hydrogen from the NADH molecule. So NADH gets oxidized, pyruvate gets reduced, and we have lactate. So this is catalyzed by lactate dehydrogenase, and then we get our oxidized NAD+ molecule again. In yeast, this is catalyzed by 2 different steps. Our final product is ethanol. So we decarboxylate pyruvate. So we release a molecule of CO2, which is catalyzed by pyruvate decarboxylase. We then have acetaldehyde as a intermediate, and then alcohol dehydrogenase is what's responsible for oxidizing our molecule of NADH back to NAD+. And our final product is ethanol. So this is how we make alcohol. All right, the reverse of glycolysis is gluconeogenesis. So we essentially make glucose out of different precursor molecules. And this usually typically occurs when our glycogen storage is low, so we don't have a lot of glycogen, and we can't really utilize glucose, so then we need to make glucose from different precursor molecules. So technically, this happens in the reverse of glycolysis. However, we need to bypass the steps that are irreversible in that process. So just like I said a couple minutes ago, some of these steps are tightly regulated, some of these steps are irreversible. So we need to bypass those enzymes and replace them with different enzymes. So our last step needs to be bypassed because that was a irreversible step. Instead of that enzyme, we have pyruvate carboxylase and pyrophosphate carboxylase. So we make oxaloacetate, and then we make phosphoenolpyruvate. And then we move on in the same way with enolase to make 2-PG, 3-PG, 1,3-BPG, and then glyceraldehyde 3-phosphate. And then we need to bypass that phosphofructokinase step because that was also, if you remember, our first committed step, a irreversible step. Instead of phosphofructokinase, we have fructose 1,6-bisphosphatase. So we have a different intermediate molecule, and then we have fructose 1,6-bisphosphate that gets split, and then we get fructose 6-phosphate. And then lastly, we need to also bypass hexokinase. So we need to somehow make glucose out of glucose 6-phosphate, and that is catalyzed by the enzyme glucose 6-phosphatase. So the net input for this reaction process for gluconeogenesis is 6 ATP and 2 NADH. And gluconeogenesis is regulated at the 1,6-bisphosphatase step. So same way as glycolysis, it is regulated at that step right here where fructose 6-phosphate, fructose 1,6-bisphosphate get interconverted. All right, next we have a glycogen synthesis. We just talked about glycogen a little bit. It is used to store energy in form of glucose. It's super important because we're taking in a lot of glucose through food. We make glucose, we can't utilize it all at once. So we need to store it somehow so we can utilize it later on. So glucose 1-phosphate is the primary building block of glycogen. Glucose gets phosphorylated via hexokinase. We get our glucose 6-phosphate molecule the same way we get it when we enter glycolysis, but instead of going through glycolysis and generating energy, we utilize phosphoglucomutase, which catalyzes the reaction of moving that phosphate group from the C6 carbon to the C1 carbon right here. So then we have our primary building block. However, we need to first activate that building block with UTP, which is kind of similar to ATP. It's also a nucleotide of some sort. And this is a irreversible step because it has a high delta G, a high change in free energy. So our UTP molecule right here gets attached to glucose 1-phosphate to the phosphate group, and then we have our UDP glucose and pyrophosphate gets released in this step. So UDP glucose is then attached to the C4 carbon of glucose at the end of a glycogen branch. So say we have a glycogen branch right here, ignore this phosphate group, we would attach it at this OH right here at the C4 carbon OH group. And then we would have a alpha 1,4 glycosidic bond, which is the bond in which all glycogen molecules are connected. Then also remember that glycogen is a highly branched molecule, which is important because it is so much easier to break down a branched molecule and make glucose available that way, because then you can have lots of branches, lots of small branches broken down that can then get further broken down. And we can make glucose available faster. So for the branching, we need transglycosylase. So after every 7 residues, it cleaves a segment and reattaches that 7th segment molecule at a glucose at the 6th carbon. So at this carbon right here, it would attach that branch of glucose and forms a alpha 1,6 glycosidic bond. And then lastly, in glycogen synthesis, glycogen synthase cannot build new glycogen molecules. It can only attach a UTP molecule to an already, well, UDP glucose, sorry, not a UTP molecule, a UDP glucose to an already pre-existing glycogen branch, but it cannot make them from scratch. So it cannot attach glucose molecules to one another. For that, we need glycogenin. It assembles the first few glucose molecules. It kind of adds them on to glycogenin, which is an enzyme itself. And then once that chain is small enough, once it's long enough and there's enough glucose molecules attached to each other, then the other molecules, so glycogen synthase, UDP, glucose, pyrophosphorylase, and so forth, take over, and the glucose chain can grow. Okay, and then we also need to be able to break down glycogen when we don't have a lot of glucose available and we need to make energy. We're either in starvation mode, or not necessarily starvation, but in low fuel mode because we haven't eaten in a while, or we're very active right now and we need a lot of energy, then we need to break down glycogen. This is done via phosphorylases, so the breakdown of bonds via the addition of a phosphate bond. So we have a glycogen molecule and we add a phosphate group to it, well, to the bond, the alpha 1,4 glycosidic bond, and we release a molecule of glucose 1-phosphate. Phosphoglucomutase, which catalyzed that same reaction in glycogen synthesis, but just the other way, then makes glucose 6-phosphate, and we can enter glycolysis. The debranching enzyme removes the alpha 1,6 linked residues of those branches via a regular hydrolysis reaction, so not a phosphorylasis, but a hydrolysis, so the addition of water, and then those branches can further be broken down in that same way via glycogen phosphorylase. And glycogenolysis mainly happens in liver cells, and glucose is then available to enter the bloodstream. Okay, and one last pathway in which glucose can be utilized is the pentose phosphate pathway. This is important because we can make a 5-carbon sugar, which can then enter and we can build ribonucleotides. So we have glucose 6-phosphate, and 6-phosphogluconolactonate is the starting molecule for the pentose phosphate pathway. So we need to somehow interconvert between glucose 6-phosphate to 6-phosphogluconolactone, which is done via the reduction of NADP, not NADH, but NADP, plus this gets reduced with that OH right here. We add that proton to NADP, we get NADPH plus, and right here we have that carbonyl bond, and we have 6-phosphogluconolactone. Then we need to open the ring and produce 6-phosphogluconolactonate with the addition of a water molecule. So we hydrate our molecule. Once we have our starting molecule, 6-phosphogluconate gluconate is oxidized and decarboxylated by 6-phosphogluconate dehydrogenase. It involves another reduction of NADPH, and we release a molecule of CO2. So we go from a C6 body to a C5 body. So now we only have 5 carbons. And if you see right here, we have that CO double bond instead of at the C1 carbon. Ribulose 5-phosphate can then be used to be interconverted to ribose 5-phosphate. So not ribulose 5-phosphate, but ribose 5-phosphate, because ribose 5-phosphate can then be converted to deoxyribose sugars with NADPH acting as a reducing agent. So NADPH gets oxidized again, it reduces ribose 5-phosphate, and then we can start building nucleotides. So this pathway is important, or an important step for building nucleotides. All right, and then to summarize all of these fun steps and how we can utilize glucose. So we have our molecule of glucose right here. We can either, if we have a lot of glucose, immediately store it. So we activated glucose 6-phosphate, we interconverted to glucose 1-phosphate, and then we can go the glycogen synthesis way. We can make glycogen with the activation of UTP, and then in reverse, if we need our glucose available again, then we can break it down via glycogenolysis. Do not get those words mixed up because I know they're very similar. You have glucogenolysis, glycogenesis, glycolysis. I know it's hard to keep track of them, but try to get that hammered in your heads, what word is for which pathway. Okay, if we want to make energy, we enter glycolysis. We generate ATP via glycolysis pathway. We make 2 ATP and 2 NADH. Our final molecule is pyruvate, and then we can either enter oxidative phosphorylation, or we can, under anaerobic conditions, make lactate, or we can start building amino acids. And then lastly, we can also enter the pentose phosphate pathway, in which our final carbon product would be a ribose 5-phosphate. All right, say we enter the citric acid cycle and we have oxygen available and we want to generate a lot of energy. Then the next fate for our pyruvate molecule would be the pyruvate dehydrogenase reaction. So pyruvate, the product from glycolysis, will ultimately be oxidized into 3 CO2 molecules. So per glucose, remember we have 2 pyruvates, so per glucose we will get 6 CO2, but per pyruvate we will get 3 CO2. And pyruvate dehydrogenase is the decarboxylation of pyruvate by pyruvate dehydrogenase, which is a multi-subunit molecule. And the overall reaction of this is pyruvate plus a coenzyme A plus a molecule of NADH, and the final product of this reaction would be an acetyl-CoA molecule, 1 CO2 molecule, and an NADH molecule. Like I already said, pyruvate dehydrogenase is a multi-unit complex that includes 3 types of enzymes. Our first one is pyruvate is decarboxylated. We release a molecule of CO2. In the enzyme 2, the leftover hydroxyl group is transferred to the enzyme E2, and then the acetyl group is transferred to coenzyme A, and E2 is restored with the help of E3. And then E3 is lastly restored as well, and NADH is reduced, and we get a molecule, well, NAD plus is reduced, and we get a molecule of NADH. So to break this further down and to look at the individual molecules, we have enzyme 1, which uses a thiamine pyrophosphate. Thiamine is also known as vitamin B, is an important cofactor for many decarboxylation reactions. So in this reaction, the phosphorylated version of thiamine pyrophosphate is needed for the decarboxylation of pyruvate. So TPP, thiamine pyrophosphate, contains an acidic proton. TPP loses its proton, and then the TPP thiazolium ring, so this ring right here, attacks the carbonyl carbon of pyruvate right here. We have a rearrangement happening, and lastly, we release CO2 right here, and we have that hydroxyethyl TPP molecule in enzyme 1 or attached to enzyme 1. In the next step, enzyme 2 uses a lipoamide cofactor. It acts as the acceptor of the hydroxyethyl group that we just saw in enzyme 1. The lipoamide group is a lipoic acid bonded to a lysine residue in the enzyme 2, and it has a disulfide bond, as we can see right here. And that disulfide bond on the lipoic acid is the active portion, which is responsible for the transfer from E1 to E2. So we attack that hydroxyethyl group and transfer it on to the lipoamide group right here. So that disulfide bond is broken, and now it carries that acetyl group. And then in the third step, enzyme 2 transfers the acetyl group from that lipoic acid portion on to a coenzyme A. So coenzyme A comes in here. It has that SH bond, that SH right here. So that sulfur attacks that carbon right here on the hydroxyethyl group. And then lastly, we have our acetyl CoA, and enzyme 2 is regenerated. In the fourth step, we need to actually restore E2. So we need to go from that oxidized state of the lipoic acid portion back to the reduced state. We need to have that disulfide bond again. And that is possible because enzyme 3 has a disulfide bond and carries a FADH group, which right now is reduced. As it gets close to that oxidized lipoic acid residue, it gets oxidized, and our E2 gets restored. We restore that disulfide bond right here. And then in the last step, step 5, we need to restore our reduced molecule in E3. We need to restore the final version of our enzyme 3 again, so it can catalyze the same reaction over again. So we bring in a molecule of NAD+, which then gets reduced to NADH, and our E3 is then restored. All right, and then we move on to the citric acid cycle, and we have a lot of more fun reactions. To be specific, we have a set of 8 reactions in which the acetyl group is condensed with oxaloacetate, a C4 molecule, and we yield a C6 molecule because our acetyl-CoA is a C2. And in the process of the citric acid cycle, we lose 2 CO2. Oxaloacetate is regenerated in our final C4 body, and each round of citric acid cycle generates 2 CO2, 3 NADH, and 1 FADH, which then gets added onto into the Q cycle, and 1 molecule of GTP or ATP. Remember, this happens twice per molecule of glucose because each molecule of glucose gets broken down into 2 pyruvate, which then gets broken down into 2 acetyl-CoA, which then enter the citric acid cycle. Many of these steps are regulated via feedback inhibition, which we will talk about once we talked about the citric acid cycle. So our first reaction is catalyzed by citrate synthase. It adds an acetyl group to oxaloacetate. Citrate synthase undergoes a conformational change, after binding to oxaloacetate to allow binding of acetyl-CoA. So it synthesizes that CC bond. The overall mechanism of citrate synthase is that oxaloacetate and acetyl-CoA bind to the enzyme, and then asparagine 375, a base in the active site of the enzyme, removes a proton from the acetyl group to produce an enolate intermediate, which is then stabilized by hydrogen bonding to histidine 274, another residue in the active site of citrate synthase. And this is also the rate limiting step of the citrate synthase reaction. The enolate then attacks oxaloacetate to produce citral-CoA intermediate, and then in hydrolysis reaction releases the CoA and the citrate molecule. So this is our first molecule in the citric acid cycle. From citrate, we then move on to reaction number 2. We have acetonitase, which is catalyzes a isomerization reaction from citrate to isocitrate via this intermediate acetonitate right here. Reaction 3 is catalyzed by isocitrate dehydrogenase, and our first molecule of CO2 gets released here, and it is our first decarboxylation reaction. So NAD plus gets reduced, we get a molecule of NADH and a rearrangement happening right here, so we can release our CO2 molecule. And we also have a cofactor, Mn2 plus, stabilizing this intermediate so that CO2 can be released right here. And our final product is alpha ketoglutarate. In reaction 4, we have our alpha ketoglutarate, and alpha ketoglutarate dehydrogenase releases the second CO2. So we release another molecule of CO2 right here. And it is a similar multi-enzyme complex to pyruvate dehydrogenase. So that fun 3-part enzyme we just talked about, it has a similar mechanism. And we have another reduction of NAD plus to NADH, and we release CO2. So our next molecule is then a C4 molecule, succinyl-CoA. What's important to note here is that the carbons that are released in CO2 are not the same carbons that enter from acetyl-CoA in that same round, but they come from the oxaloacetate, which was regenerated in previous rounds of the TCA cycle. Reaction 5 is succinyl-CoA synthetase, which catalyzes a substrate level phosphorylation. So our substrate gets phosphorylated, hence the name. The phosphate group displaces our coenzyme A in succinyl coenzyme A to produce succinyl phosphate, an intermediate. And that phosphoryl group then gets donated to a histidine residue on the enzyme itself, and the phosphoryl group is then transferred to GDP to form a first molecule of GTP or ATP. So we produce some energy here. And our molecule that comes out of this is a C4 molecule, succinate. Then we start regenerating oxaloacetate. So in the next step, we have succinate dehydrogenase, which generates a molecule of ubiquinol. So we go from succinate, which gets oxidized, and we reduce the enzyme which carries an FADH cofactor. That FADH gets reduced into FADH2, and FADH2 then donates its electrons to ubiquinol, and ubiquinone is required to take the electrons from FADH2 and then pretty much immediately enters the electron transport chain from here on. So remember succinate dehydrogenase when we talk about the electron transport chain. All right, reaction number 7 is fumarase, catalyzed by fumarase. It's a hydration reaction to form malate. This is the last precursor before we regenerate our oxaloacetate, which is catalyzed by malate dehydrogenase. It reduces another molecule of NAD plus to NADH, and we get our final molecule of oxaloacetate, so we can start over another round of the citric acid cycle. To quickly look at the thermodynamics of glycolysis and the citric acid cycle. So the citric acid cycle is energy generating, but only if we enter oxidative phosphorylation after this. So only under aerobic conditions do we actually produce energy. So if we enter oxidative phosphorylation, all those cofactors that we just produced during the TCA cycle, so the 6-NADH and 2-ubiquinol and the ATP that was generated, or GTP that was generated, is then used to produce ATP. Well, not the GTP itself, but the NADH and the ubiquinol. So under anaerobic conditions, none of this can actually happen because we do require oxygen, hence the name oxidative phosphorylation. And the only ATP we generate under anaerobic conditions happens during glycolysis, where we generate that 2-ATP directly. None of these other molecules will be generated. However, if we have our aerobic conditions, then we can generate up to 32 molecules of ATP. So oxidation of each NADH molecule yields 2.5 ATP, and oxidation of FADH, which is right here, already transferred its electrons to ubiquinol, generates 1.5 ATP. So the full output of a full cycle of oxidative phosphorylation can yield 32 molecules of ATP. All right, like I already said, the citric acid cycle is tightly regulated at all those steps that I already mentioned. So at the 3 irreversible steps, one of them was citrate synthase, isocitrate synthase, alpha ketoglutarate dehydrogenase. Those are regulated via feedback inhibition, in which if we have a lot of NADH, so we already have a lot of those cofactors, we just ran through the citric acid cycle a bunch of times, we do not need to make any more. That will be a waste of energy, a waste of resources, essentially. So that NADH then signals, hey, there's a lot of us, don't make any more. And it reversibly inhibits those enzymes until there's not a lot of NADH left anymore. And then the citric acid cycle can resume. On the other hand, if there's a lot of calcium and a lot of ADP, so not ATP, but ADP, those act as activators, and they activate isocitrate synthase and alpha ketoglutarate dehydrogenase to produce more of those cofactors. All right, and one last characteristic of the citric acid cycle is that many of the citric acid cycle intermediates are precursors for other molecules. 6 of the 8 molecules used in the citric acid cycle can be used in other pathways as well. For example, glutamate is a precursor for glutamine, arginine, and proline, and can also be used as a precursor for purine and pyrimidine nucleotides, and it can be synthesized from alpha ketoglutarate. Citrate can be used for fatty acids and cholesterol. Malate can be converted into pyruvate. Oxaloacetate can be used with pyruvate to make glucose. Succinyl-CoA is important for the heme group. So lots of these molecules can be utilized in other ways. Also important to know about the citric acid cycle is that it is called anaplerotic reactions. So the citric acid cycle can be replenished through those same molecules. So we can fuel in intermediates into different steps of the pathway to replenish, for example, oxaloacetate, alpha-ketoglutarate, succinyl-CoA, and fumarate. So in the same way that we can use different molecules as precursors, we can also fuel in different molecules. All right, moving on to oxidative phosphorylation. Now it gets really fun and complicated. So oxidative phosphorylation, as the name suggests, requires oxygen. Electrons are extracted and passed on through the electron transport chain to a terminal electron acceptor. Movement of electrons through the electron transport chain generates energy that will be used to fuel the proton pump, and it generates a proton gradient. And the proton pump energy is then used to synthesize ATP from ADP and phosphate, and it converts energy from the proton gradient that is then going to be stored in form of a chemical bond. Just as a quick refresher, if you remember oxidation and reduction principles, oxidation is the loss of electrons, reduction is the gain of electrons. I know this seems kind of counterintuitive, but if you think about oxidation numbers, in reduction, as we gain electrons, the oxidation number gets reduced, hence why it's called reduction. The reduction potential is the measure of a substance or the tendency of a substance to gain electrons, so to get reduced. And the greater the value, the higher the tendency to accept electrons. So if I have a high reduction potential, then I am very likely to accept electrons. So usually electrons move from a molecule with a low reduction potential to a molecule with a high reduction potential. If you remember all the fun math that we did for exam 2 with the Nernst equation, remember to take into account the number of electrons to solve that equation, so how many electrons are moving along. And the free energy change can be calculated from the change in reduction potential from donor and acceptor. And the larger the change in free energy, the higher the tendency of electrons to flow from one molecule from one side to another. So if we look at our electron transport chain, we transport electrons from NADH, from the citric acid cycle, and from glycolysis to complex 1, to complex 3, which then gets funneled into cytochrome C, and cytochrome C then transports electrons to complex 4, and lastly to the proton pump, where we then generate ATP and a molecule of water. The electron transport chain takes place in the mitochondrion. The mitochondrion has 2 membranes, a outer and a inner membrane, and the inner membrane is folded to increase the membrane space. The folds are called cristae. So we need a lot of membrane space because then we can have more proton pumps. So increasing our membrane space by folding it makes a lot of sense. So the electron transport chain happens along the inner membrane, and the TCA cycle occurs in the matrix and the intermembrane space. Glycolysis, as a reminder, happens in the cytosol, and electrons get carried from all those reactions and get funneled around, essentially. Therefore, we need transport systems to bring molecules across the mitochondrial membrane, either into the mitochondrion or out of the mitochondrion. For example, we have the malate aspartate shuttle, which transports essentially oxaloacetate, but oxaloacetate cannot cross the membrane itself. So it needs to be converted to malate in the cytosol, which then malate can pass the mitochondrial membranes into the matrix, where it can then be part of the electron transport chain, and malate is then used and regenerated back to oxaloacetate, which can then be converted to aspartate, and then aspartate can be transported back out of the mitochondrion back into the cytosol and reconverted into oxaloacetate. So that's just one example of one of those transport mechanisms. All right, so taking a look at the electron transport chain, we have 4 different protein complexes, 1, 2, 3, and 4. These are not actually named in the order in which they happened, but the order in which they were discovered. So we have complex 1, in which electrons from NADH get funneled to ubiquinone. In complex 2, we have electrons from FADH. If you remember, we just talked about this, succinate dehydrogenase. They get carried to Q, and then we get our ubiquinol, that Q, which then carries electrons to complex 3, where ubiquinol then transports a proton to cytochrome C, this little guy right here, and cytochrome C then carries electrons to complex 4. And as electrons get transported through each of those complexes, 1, 3, and 4, not complex 2, just 1, 3, and 4, protons get transported from the matrix into the intermembrane space. So we have more protons, a lower pH in the intermembrane space than we do in the matrix. All right. Complex 1, has a iron sulfur cluster. 2 electrons are donated from NADH. That are then picked up and transported to ubiquinone, and 4 protons are pumped and transported across the membrane. Just as a hint, this is flipped to the image that we just saw earlier. So the matrix is now here, and the intermembrane space is right here. In the previous picture, it was the other way around, so don't get that mixed up. We're still pumping electrons from the matrix into the intermembrane space. So to summarize, NADH drops off 2 electrons. They travel from a flavin molecule to the iron sulfur cluster until they reach ubiquinone to form ubiquinol, that QH2. Ubiquinone, however, can accept electrons from complex 1 and complex 2, acetyl-CoA dehydrogenase and mitochondrial dehydrogenase. However, only complex 1 contributes to the proton gradient. So even though complex 2 also gives electrons to ubiquinone to form ubiquinol, only complex 1 shuttles protons. All right, complex 3 then transfers electrons from ubiquinol, so from that QH2 to cytochrome C. Cytochromes are proteins that contain heme groups. I already mentioned heme groups a little bit earlier, but if you remember from hemoglobin, they have a iron in their center, an Fa3+, in the middle of the porphyrin ring, and that iron can be reduced from Fa3+ to Fa2+. In complex 3, we have something happening called the Q cycle, which is honestly a little bit complicated and can be really confusing. But essentially, the Q cycle only takes 1 electron at a time, but 2 electrons get donated from ubiquinol, so we must split it up to pass each electron through a 1 electron carrier. So that cycle is going to be repeated for each molecule of ubiquinol, and then each cycle is repeated 2 times again. So the overall reaction is we have 2 ubiquinols dropping off 2 electrons each. So we have 4 electrons. And then those electrons get transferred onto 2 cytochrome C. Iron 3+ gets reduced to iron 2+. So in the end, we have 1 molecule of ubiquinol, 1 ubiquinone, and 2 cytochrome C with a reduced iron. So for 2 molecules of ubiquinol that drop off 2 electrons, 2 cytochrome C get produced, and 4 protons will be transferred to the intermembrane space. So from the matrix pumped into the intermembrane space. All right, I know this is a lot of information, but remember that protons from matrix to intermembrane space, and the electrons get passed on from a lower reduction potential to a higher reduction potential. So now we are at cytochrome C. And then cytochrome C carries our electrons from complex 3 to complex 4. And then complex 4 oxidizes cytochrome C. So it takes the electrons and reduces O2. And remember, each cytochrome only carries 1 electron each. So we need multiple cytochrome Cs. So we need 2 cytochrome Cs that drop off their electrons for each molecule of H2O that is produced. And for each molecule of H2 that is produced, 2 protons will be transferred to the intermembrane space. So complex 4, or 2 cytochrome C molecules, get oxidized again. We get our regenerated cytochrome C that can then go back to complex 3. We pump 2 protons from the matrix into the intermembrane space. Our 2 electrons from cytochrome C get transported along complex 4, and from a molecule of O2, that's going to be reduced, and we get a molecule of H2O. So now we have all this energy that was generated from the electron transport. Complex 1, that reduction of NADH to ubiquinol, we will yield about 69 kilojoules per mole. In complex 3, ubiquinol to cytochrome C, about 36 kilojoules per mole. And in complex 4, cytochrome C to O2, about 120 kilojoules per mole. And all of this is for every NADH that enters the electron transport chain. So for each NADH entering the electron transport chain, we get so much free energy. So now we need to do something with this energy, which this is coupled to proton transport across the membrane. So we already talked about this. Each of the complexes shuttles protons from the membrane into the intermembrane space. So now we generated this proton gradient. So now we gotta do something with it. So the protons don't like that there's so many protons here and not as many protons here, and they want to go back. So they have that drive to equal out that gradient. So they're gonna find a way to get back in there through the ATP synthase, which is also called the protomotive force. So the ATP synthase consists of multiple subunits. We have the F1 subunit, which is a transmembrane protein, so that sits in the inner membrane and allows for flow of protons from the intermembrane space back into the matrix. And the F1 subunit right here forms ATP through the movement of these alpha and beta subunit. The F0 subunit is a ring consisting of 8 C subunits. You can see them all right here. And this subunit right here that is attached to the F1 ring to kind of stabilize it. Our F1 ring has 3 alpha and 3 beta subunits surrounding a gamma shaft. The F0 ring rotates around the subunit as proteins enter right here from the intermembrane space. They then bind to the C subunits, and the C subunit rotation is driven by electrostatic attractions and repulsions. So as the rotation continues, the electrostatic attraction kind of goes away, and the subunit moves away from right here. And per rotation, as a proton moves in here and away from the subunit, another subunit comes in here and a proton gets released into the matrix. The F1 component is responsible for ATP synthesis, so the alpha and beta subunits. However, the subunits themselves don't rotate, but this gamma shaft right here does rotate. And as that shaft rotates, it kind of bumps along these alpha and beta subunits, and that mechanical energy then gets transferred into chemical energy, so into the bond of ADP and pyrophosphate to form ATP. So we have this binding change mechanism. We have 3 different conformational states. Per subunit, we have a loose state in which ADP and phosphate bind. We have a tight state, which catalyzes the bond or the reaction from ADP and phosphate to ATP, and then the open conformation, which releases ATP. So one cycle requires 3 rotations of that gamma shaft, in which that rotation then bumps into these subunits and changes those conformations. And 3 ATP molecules are made for every 8 protons that are pumped through the F0 subunit. So if you break that down, then 2.7 protons are needed for each ATP that is produced. So for one NADH molecule, 10 protons are released, and 10 protons then yield 2.5 ATP. So hence why we say one NADH yields about 2.5 ATP. Like I said, I know this is a lot. If you need to go back and kind of look at all this again, please do. But all of this is connected. And if you get where those connections come in and how all of those steps kind of flow into each other, I think it makes a lot of sense. So to summarize all of this, glycolysis breaks down glucose, pyruvate is turned into acetyl-CoA. Acetyl-CoA then enters the TCA cycle. We have our reduced cofactors that enter the electron transport chain. We have one NADH that produces 2.5 ATP per NADH, and we have one ubiquinol that produces 1.5 ATP per molecule. So overall, oxidative phosphorylation glycolysis, under aerobic conditions, we can produce up to 32 molecules of ATP. All right, we are almost at the end, so bear with me. Okay, so let's put all of this into context. We have different metabolic pathways that can happen in different cellular locations, all at different times. So the TCA cycle, oxidative phosphorylation, both happen in the matrix. Glycolysis, glyconeogenesis, and the pentose phosphate pathway happen in the cytosol. So now let's look at the different organs and tissues and how those pathways function there. So imagine you just ate something, you are well fed, you have a high glucose level in your bloodstream. So that means in your liver, glucose enters your liver. You have a lot of glucose, so you're gonna store glucose in form of glycogen. We're also gonna enter glycolysis. We're gonna have acetyl-CoA as an outcome, which can then be used to build dry acylglycerols, which can then enter into the bloodstream again. And then we can make fat cells. If we are in a fed state, we can also take in amino acids, which can then be turned into urea, or we have lactate, which can then be turned into glucose. This can both happen in a fed and a fasted state. In our muscle cells, if we are fed, we have a lot of glucose, and we don't need to convert that glucose right now. We're not active. Then also in our muscles, we're gonna build glycogen. If we are in a fasted state, so about 12 to 18 hours after eating, our glucose levels are starting to get low. In the liver, that means we're gonna start breaking down glycogen and amino acids to then have glucose readily available, which can then enter the bloodstream. Dry acylglycerols in the liver can then be turned into acetyl-CoA and ketone bodies, and they can also enter the bloodstream. All right, in adipose tissue, sorry, I forgot to mention that in the fed state, also when we have glucose available, we're also gonna then have glycerol available, and we have our fatty acids, which can then be turned into dry acylglycerols, and we're gonna start having fat cells that will be stored. In a fasted state, we have our triacylglycerols, and we need to make them available. So we're going to start breaking them down into fatty acids again, and they will enter the bloodstream. In starvation mode, meaning we haven't eaten in more than 48 hours, don't recommend doing that. We have low to no glucose. So that means that we are starving and our muscles will start breaking down protein, will start breaking them down into amino acids, and those amino acids will then be made available in the bloodstream for further breakdown, which not ideal. Don't do that. All right, so our metabolic pathways, oh, sorry, I forgot the active state. In the active state in the muscles, we have lots of glucose available, either from glycogen that we made earlier when we were in the fed state, or from glucose entering in the bloodstream. And if we are super duper active, then we have a very high energy demand and not a lot of oxygen, which means we're very likely to enter lactic acid fermentation, and we're going to start breaking down our pyruvate into lactate instead. All right, so our metabolic pathways, they are circuits and depend on inter-organ transport. So they need to get from one organ into another one. For example, from liver to muscle. So we need to be able to transport glucose from the liver to the muscle. So then glucose can get broken down to lactate, and then that lactate enters the bloodstream again and gets built in the liver. Right here it comes in and gets rebuilt into glucose via gluconeogenesis. And that is called the Cori cycle. And then we can also transport nitrogen from muscle to liver. So in the same way, when we break down pyruvate via anaerobic breakdown, we produce alanine, and that is going to be transported via the bloodstream into the liver, and then gets recycled to pyruvate, and urea is going to be excreted from the liver. All right, and then we also need to be able to control our fuel metabolism, and hormones are usually responsible for this. Hormones are chemical messengers that are secreted by specialized tissues into the blood, and they trigger a physical response in certain cells and organs that carry certain receptors that those hormones can bind to. So only if the cell actually carries the receptor will it respond to that hormone. Hormones are effective at really low concentrations. However, hormonal control is pretty slow because the chemicals moving through the bloodstream. Our fuel metabolism hormones are insulin and glycagon. Insulin is a water-soluble peptide hormone that signals high glucose, and glucagon is also a water-soluble peptide hormone that signals low glucose. Glucagon and insulin are released by islet cells. Alpha cells synthesize insulin, and beta cells synthesize glucagon. And hormone production and release are both regulated by exocrine cells that sense blood glucose levels. And then we also have epinephrine and norepinephrine. That should sound familiar when you think about fight or flight response. They are both water-soluble monoamine hormones and are triggered under low glucose and on a fight or flight response. Glucose levels are detected by glucokinase. It acts as a cellular sensor and is specific to glucose, but has lower affinity glucose than, for example, hexokinase, which makes sense because we want hexokinase to bind glucose, especially when we have a lot of glucose present. Okay, so let's take a look at this right here. Insulin promotes glucose transport into cells because we have high glucose. We don't need all of that glucose sitting in the bloodstream. So it stimulates glycogen synthesis and suppresses glycogen breakdown in muscle. In adipose tissue, insulin activates extracellular lipoprotein lipase, increases levels of acetyl-CoA carboxylase, and stimulates triacylglycerol synthesis, and also suppresses lipolysis, because again, We don't need to make more glucose available. We want to store it. In the liver, it promotes glycogen synthesis and promotes triacylglycerol synthesis as well, and suppresses gluconeogenesis. So also increased insulin stimulates addition of glutamine-4 transporters, so transporters that are responsible for uptake of glucose into the cells. So we have high glucose, we have insulin. Insulin is a phosphatase, like insulin activates phosphatases. That phosphatase dephosphorylates glycogen phosphorylase A, which then turns into its inactive state, glycogen phosphorylase B, because we do not want to break down glycogen. On the other side, insulin activating phosphatases will dephosphorylate glycogen synthase B, which is less active, and as it gets phosphorylated, it activates, or it's going to turn into its active state, glycogen synthase A. It's going to be more active. We're going to produce glycogen. In turn, if we have low glucose and we have like glucagon signaling low glucose, it's going to activate a bunch of different kinases, and those kinases are then going to phosphorylate glycogen phosphorylase B in its inactive state. And as it's going to be phosphorylated, it's going to be in its active state, glycogen phosphorylase A. Glycogen breakdown will be stimulated. We will break down glycogen. In turn, glycogen synthase B, that is phosphorylated, that is glycogen synthase A, which is dephosphorylated, will get phosphorylated by the kinase, and we will have glycogen synthase B, which is less active. Glycogen production will be slowed down. So glucagon will stimulate glucose mobilization in liver and lipolysis in adipose tissue. We need to have glucose made available for us. And then lastly, talking about metabolism disorders and different sorts of fat tissues. We have white fat and brown fat tissues. White fat is visceral and subcutaneous fat, so it is pretty much the fat cells that we in normal adults produce. Brown fat is actually brownish, has a little brownish tint, and it gets its color from mitochondrial enzymes and blood vessels, and is an adipocyte with multiple lipid droplets inside of it. It can generate heat. The breakdown of brown fat generates heat, and it is commonly seen in infants because they need to stay warm. All right. A very common metabolism disorder is diabetes, which is characterized by hyperglycemia, high levels of glucose. There's two different types. We have type 1, which is an autoimmune disease. It destroys insulin-producing cells, so our body cannot produce insulin itself anymore. We need insulin injections because we cannot make it on our own anymore. Type 2 diabetes, we have normal insulin levels. Our body can make it the normal way by ourselves. However, our cells are resistant to insulin, so responding cells don't respond to it anymore, and we need to have specific diets and exercise regimen. So in diabetes overall, the tissues lose ability to respond or produce insulin, resulting in hyperglycemia, so high glucose all the time, which can be really dangerous because glucose then gets converted to sorbitol, which can then alter the osmotic balance of cells. So the water intake, uptake, and loss of cells will be altered. And then lastly, we have metabolic syndrome, which can be a cluster of conditions that occur together and can increase risk of type 2 diabetes and different heart diseases. Well, heart disease, but caused by different diseases. And that is characterized by increased blood pressure, high blood sugar, excess body fat around the waist, and abnormal cholesterol levels. All right, I think this covers pretty much everything that should be important for exam 4. I hope this was somewhat helpful and put all of the information into context. again, feel free to reach out to me if you have any questions. If you struggle to understand any concepts, I'm more than happy to help. This is, for many of you, hopefully the last exam in this class. And remember that if you get a good grade, you might not have to take your final, depending on which section you're in. But yeah, make sure you study hard and grind for the rest of the semester. And good luck with studying.